Hello and welcome to this talk about the Orthoptera, the insect order commonly known as grasshoppers, of the Lower Cretaceous CRATO Formation of Brazil. I am Ole Schall, a German PhD student from Hamburg, Germany. This year, I have had the opportunity to study a plethora of specimens of fossil grasshoppers in a collaboration with Dr. Daniel Lima and Professor Dr. Alison Pinheiro from the Museo de Paleontologia Plácido Cidade Nuvens, Dr. Sam Hatz from the University of Illinois, Dr. Ulrich Kotthoff from the Leibniz Institute for the Analysis of Biodiversity Change in Hamburg, and Professor Dr. Martin Husemann from the Public Museum of Natural History in Karlsruhe, Germany. To all these people, I wish to express my warm thank you for exploring this exciting subject along with me. In the present talk, I want to give you an introduction to the grasshoppers of the CRATO Formation. First, I am going to give you an idea about the time and space that the CRATO Formation was deposited, as well as illustrate briefly the paleo-environment the Orthoptera inhabited. The main part then is focused on making you familiar with the different groups of grasshoppers that have occurred in the formation. You will learn how to identify the most commonly encountered families and get to know some interesting facts about them. Lastly, I will address the importance of the CRATO Formation to understand the evolution of Orthoptera on a larger scope. The contents of this talk are largely based on 4 studies I wrote as part of my PhD. The first of them was published only about 2 weeks ago in the journal Zootaxa. Before we dive into the world of CRATO Formation grasshoppers, here are just a few words about myself. I studied biology at the University of Hamburg in Germany. Because I had already written my bachelor's and master's thesis about fossil grasshoppers, I continued the academic pathway in paleontology at University of Hamburg and Leibniz Institute for the Analysis of Biodiversity Change. I am interested in everything related to fossil Orthoptera, but my main groups of focus are the Echinidae, Leucostopsidae, and Tridactyloidea. Now, let's travel back to the Lower Cretaceous of Brazil and meet the jumping insects of the CRATO Formation. The CRATO Formation is part of the Santana group in the Araripe Basin in northeastern Brazil, and widely recognized as one of the most important Konzentratlagerstätten of the Lower Cretaceous in the world. Although debated, the CRATO Formation is mostly considered to be of Aptian age, circa 125 to 113 million years ago. The deposits themselves probably represent a shallow, intermittently hypersaline lacustrine system under predominantly high-stress conditions, such as low oxygen or elevated salinity levels. While some authors believe the CRATO Formation was once an extensive freshwater lake, others consider it a lagoon system. Surrounding the water body was probably a wetland environment in a semi-arid climate with extensive and long-lasting marshes. The fossil assemblage found in the CRATO Formation is very diverse. The flora is made up of water plants, gnatales, conifers, ferns, and early angiosperms. Among vertebrates, you can find fishes, amphibians, reptiles, avian and non-avian dinosaurs, as well as pterosaurs. Arthropods and insects are especially diverse, earning the formation a reputation as one of the richest and most important Cretaceous insect localities worldwide. One of the most commonly encountered families of grasshoppers in the CRATO Formation are the members of the Leucostopsidae, to which I like to refer as lakehoppers, although this name has not been officially proposed yet. Orthoptera are split into 2 suborders, the Calliphera, which includes grasshoppers, pygmy mole crickets and their relatives, and the Ensifera, which includes katydids, crickets, and their relatives. The lakehoppers are part of the suborder Calliphera. There are 3 subfamilies currently recognized, all of which can be found in the CRATO Formation. In these subfamilies, there are currently 11 described genera and 26 species from CRATO. A problem with fossil Calliphera, and Leucostopsidae in particular, is that the apomorphies for identification are difficult to determine. In extant Calliphera, apomorphies are often based on the morphology of the internal genitalia or molecular findings. Indeed, apomorphies for Leucostopsidae are still yet to be defined, even more than 100 years after their first description. That being said, to tell if a specimen of fossil grasshopper from the CRATO Formation belongs to this family, you can check for a couple of characters. Lakehoppers have antennae that are shorter than their bodies. Their forewings distinctly surpass the abdomen, and the ovipositor of the females is short, with the valvulae often broad and serrated. In addition, there are possible diagnostic features in the venation of the forewing. Lakehoppers are the most common members of Calliphera in the CRATO Formation, both in terms of diversity as well as abundance. Some lakehoppers, like the recently described species ARARIPE locusta imperatrix, had curious head appendages which, based on their position above the eye, are unlikely to be part of the antenna. Their function, for now, is unknown. Another family often found in the CRATO Formation, however significantly less diverse in terms of species number than the Locustopsidae, are the lagoon crickets or Elcanidae. These insects belong to the other suborder, Ensifera. They are characterized by 3 pairs of large movable spurs on the dorsal side of their hind tibiae. Additionally, they have antennae much longer than their body, and usually a long and prominent ovipositor. In 2025, Wilmot et al. Recognized 3 types of ovipositors present in lagoon crickets. One that looks like a sword, one that looks like a scythe, and one that resembles a small saber. The saber type is rather small and delicate, and for this reason can be tricky to identify. The females of one species of lagoon crickets found in the CRATO Formation, Mesembrelcana rasnezini, were long thought to be males of another species, because their saber-type ovipositor got overlooked. Another group that is both diverse and numerous in the CRATO Formation is the Grilloidea, commonly known as crickets. The crickets of the CRATO Formation belong to 2 different families, Grillidae and the extinct Baisogrillidae. The main point of differentiation is the orientation of the crossveins of the mirror, a specialized structure in the forewing of male Grillidae and Baisogrillidae. Crickets can be recognized based on their long antennae and a prominent, sometimes very long and needle-shaped ovipositor. Because of the issue that fossil female crickets cannot be placed below the superfamily level due to the reliance on male forewing venation, Martin Sneto and Vastasi in 2009 erected 2 artificial genera to house female specimens that cannot be placed into one of the male-based genera for now. A curious little taxon of Orthoptera found in the CRATO Formation are the monkey hoppers, or Eimostacoidea. These califerans may not have been as diverse as their relatives in the Locustopsidae, but their rarity in the fossil record makes every single species ever so more important for unriddling the puzzle of Orthoptera evolution. Eimostacoidea can be differentiated from the similarly looking Locustopsidae by the fusion of 2 veins in the forewing, which are not fused in lakehoppers. Since this year, Eimostacoidea are also known from Middle Cretaceous Myanmar amber, which is circa 10 million years younger than the CRATO Formation. Curiously, the monkey hoppers from Myanmar amber resemble their extant counterparts quite well, whereas the CRATO Formation Eimostacoidea bear high likeness to the Locustopsidae. Aside from the 4 groups already presented, there are additional members of Orthoptera present in the CRATO Formation. Not much is known about these critters, but their discovery has important implications for the evolution of their respective parent taxon. For example, the dune cricket Braugmannia gröninge and the stick grasshopper Eoproscopia martilli are not only the oldest, but also the first fossil representatives of their family discovered so far. Now, why exactly are the grasshoppers of the CRATO Formation so interesting? Firstly, because of their incredible diversity. The CRATO Formation has revealed one of the most diverse assemblages of the suborder Calliphera of the Cretaceous. In the case of the Locustopsidae, it is the most diverse fossil site in the world, with 1/3 of the species described from this location. This diversity is even more remarkable because the Aptian age of the CRATO Formation represents a key stage in grasshopper and insect evolution, but more on this in a second. Further, The CRATO formation is interesting in a paleogeographic context with other Cretaceous insect Lagerstätten, especially the earlier mentioned Burmese amber from Myanmar. Many grasshopper taxa found in the CRATO formation are also present in the amber, for example Tridactyloidea, Eumastacoidea, or the Elcanidae, of which you can see a specimen from CRATO and Myanmar amber in the bottom images. Since Myanmar amber is circa 10 million years younger than the CRATO formation, comparison of the different forms from either location allows us to follow their evolution during a key stage in insect evolutionary history, when, in the Lower Cretaceous, in an interval from circa 125 to 100 million years ago, insects reached an all-time high in diversity levels, and the angiosperm terrestrial revolution unfolded and changed the terrestrial ecosystems and life on land towards the path we know today. For Orthoptera, these changes in ecosystem, which next to the angiosperm terrestrial revolution were also driven by other factors, are probably reflected in the faunal composition of grasshoppers in the Cretaceous compared to the Cenozoic. Specifically, a major change or transitionary phase seems to have taken place during some interval between the Middle Cretaceous and early Cenozoic. Diverse Mesozoic lineages, like the Elcanidae and Leucostopsidae, disappear from the fossil record after the Cretaceous. Instead, the extant superfamilies Acridoidea and Tetigonioidea begin to dominate the orthopteran community. Curiously, the Grylloidea, although some groups like the Baisogrylidae did not survive the Cretaceous, overall maintained their diversity levels almost unchanged since their first occurrence in the Jurassic. Because of its diversity, as well as the time window in Earth's history it preserved, the CRATO formation of Brazil offers a one-of-a-kind chance to study the faunal transition that took place in Orthoptera and eventually brought forth our present-day night-singing meadow musicians right where it started. Before we leave, I have an overview of the studies I authored myself with the help of other expert researchers, as well as the ones I did not have a hand in that are worth your read if this topic has caught your interest. And with that I have reached the end of my talk. I hope this presentation was interesting to you. If you would like to get involved into the research of fossil grasshoppers too, there are plenty of opportunities, species to describe, and discoveries to be made. Thank you for your interest, and have a fantastic rest of your day.